Welcome, everyone. We're thrilled to be back in the new year for the molecular ML reading group. This is sponsored by Rosetta, and we've had a wonderful series of speakers on different deep learning models, especially diffusion models. Today, we're absolutely thrilled to have Deniz from the Kortemme group come and tell us about structure-conditioned masked language models and how they generalize beyond the native sequence space. We'll let Deniz take it away.
Thank you. Yeah, thank you, Nick and Matt, for hosting me as well. So I'll just get started. So fixed backbone design as a challenge has been around for many years and has also been addressed by many different methods. So these methods aim to sample a set of optimal sequences for a given input backbone. And so both by physics-based modeling and machine learning-based methods, it was possible to design sequences and obtain, the fixed backbone level type for various applications. So with machine learning methods in specific, they learn to approximate a probability of sequence-given structure, and there are impressively good results out there. So I'll talk about a popular method, and this is from John Ingram and others from 2019, where the model is called Structure Transformer, and it is an encoder-decoder. And so for this workflow, essentially, there are node-level informations. So residue type information you would get from the input backbone, as well as distances between residues or edge level information. And structure is encoded in terms of GNNs. So based off of learning from a graph or a protein graph specifically, the goal is to go from the structural information to a sequence that the model decodes autoaggressively. So in this case, for this model, it was a N-terminal to all C-terminal decoding order model. So it would start decoding from the first residue, terminally speaking. And in the very beginning, you can imagine that no sequence has been decoded. So there's only structural information flow in the local environment of the GNN, basically. And then as sequence information is decoded autoaggressively going through, there is some sequence information from the model that is used, as well as structural information within that local neighborhood to sort of figure out what's supposed to essentially be here. And you can imagine that for this type of model, which is autoaggressive, and the same is true for protein MPNN, which has a random decoding order, compared to the N to C. What they do is, or for a fixed backbone, what they do is given a backbone, predict the sequence, and then in doing so, since there are decoding steps, it essentially decodes one residue at a time. So if you look at the random decoding order, it would look something like this, jumping all over the place in terms of the graph. And then you can imagine maybe there are N total steps. So this would be the sequence length is N, or there are N total residues. And so in these types of models, for the encoder-decoder architecture, there's an encoder block, decoder block, and in the encoder block, this is where the structural level information is embedded. And then in the decoder, you're going to have a sort of a preceding context in terms of what the model is designing sort of one residue at a time. So you can imagine that the encoder block for these types of models is called once because the structure itself is not, as mentioned, it's fixed backbone design. And then for the decoder block, it's called the total residue count number of times. So this, you can sort of already intuit the sequential order, but for contrasting this to encoder-only models, where you have you only have an encoder block, which tends to have a lot more depth and is only called once. you have information flow bidirectional, and you're predicting missing residues from surrounding context, and doing this essentially in a single pass. So compared to this autoaggressive flow, this non-autoaggressive type of models are formulated to predict the sequence from a single pass. So you can imagine it looks like this, with a single decoding step, though you don't actually have a decoder. And so with that as background, I'll introduce Frame2seq, which is an IPA-based masked language model with state-of-the-art accuracy and speed. So once again, the goal is, if given a backbone, can we design sequences or can we predict sequences with accuracy? And in doing so, I'll talk about some of the different things we've looked at. To give away the top of the line numbers and to motivate the rest of the talk, over the held out test set, when compared to a protein MPNN model version that's trained on the same splits as Frame2seq, we see that it is Frame2seq improves over sequence recovery about 2%. And we also see an implication between the autoaggressive and non-autoaggressive formulation in terms of inference speed, especially when utilizing GPUs. So what does the model actually do, and how does it find these results? Once again, protein backbone as input. And from protein backbone, we have backbone rotations and translations. So using a Gram-Schmidt process employed by a lot of other people as well, we can construct coordinate frames. We also have backbone torsions as information or feature from the backbone, as well as sometimes partial sequence. So this part is done to train the model to have fixed positions that you might use protein and MPNN for. So whenever you have a, let's say you have a binding site that you'd like to keep constant in your protein and you don't want that designed, the way, for example, protein MPNN does that is it's trained with some understanding of partial sequence information as well. So Frame2seq can also do this and fix certain positions by training with this same idea, but otherwise, backbone torsions, possibly sequence, make up the sequence embedding. And then in terms of edge level information, there is inter-residue distances and relative positions. So that will be the pairwise embedding. So all of this goes through the first layer of Frame2seq. So Frame2seq is essentially a set of node updates and edge updates, where the node updates themselves are IPA or invariant point attention based, which is from AlphaFold2 structure module. So the first layer is an IPA layer, followed by a transition layer that is also adapted from, once again, the structure module. And based on this, the updated nodes and the edges coming from here go through an edge update. And this stack of layers essentially is followed a total of 7 times, making the bulk of the model's depth. And then at the final layer, there is another IPA, and then transition, and then projection to sequence dimension. So at this point, just like many other models of similar nature, you actually have a distribution over the amino acid types at each of these positions, and you're sampling from that. So one way to sort of change some things about sampling, or one parameter, is the sampling temperature, where you can add a little bit of noise and get different sequences from the same essentially distribution. So one thing that's different in the preprint is, well, it says per residue scores here, and I'll talk about scoring with Frame2seq in a little bit, but also for the IPA layers, it says that they're regularized. So I'll go through the IPA regularization, which sometimes in the slides is called IPA masking, in a little bit of detail. So I thought I would appeal to people from multiple backgrounds. If you look at it in terms of pseudocode and try to compare to vanilla IPA, and it might be easier to do this side by side, but essentially before attention becomes attention and goes through softmax in IPA code or pseudocode, we've written a dropout, and we found this to be most successful at 20% before the softmax. And to explain this in my favorite way over a structure, I'll just show this, the Nova Design Rossmann fold, which is a beta alpha repeating protein. And if we think of what distance information would look like for this, so if you looked at some type of distance map or what normal attention would look like based off of distances, and let's say we have n total residues, so it's an n by n map, what we would see are some signature patterns corresponding to the secondary structural units. So you can see the helical patterning here, and you also have longer range contacts, and these suggest whether you're having parallel contacts in terms of the sheets or anti-parallel, etc. And what we find, attention, with the dropout I mentioned, is that because there's less capacity to essentially distribute attention, the model has to learn from a restricted capacity, but still retain the same information since it's trained for the same objective and by the same loss term. So looking at this, we can see that in a sparser sense, it still keeps track of the longer range contacts, and we can still tell helices apart. It's just there is less redundancy in how, in terms of the patterns, it can tease apart from the structure what it's keeping track of. And this 20% is randomly dropped as well. So it has to generally learn from that restricted context. And yeah, so in terms of what it looks like for a structure, here for attention, basically the bluer it is, that residue is paid more attention. And then in terms of the side chains, they're only shown if that residue has attention greater than 0, and then this bright green color is for the attending residue itself. So this is one example of what the IPA regularization results in. But essentially, let's say that the residue here is masked, then the question becomes from what it can see, and in terms of the capacity to pay attention is not masked, still informs the model in terms of what residue identity is the best prediction. So this is a quick movie, and this is once IPA regularization has been implemented, what it would look like. And I think that for helices, it is picking up on an expected pattern, which sort of corresponds to the periodicity. And in terms of the beta sheets, so whenever it's designing a beta sheet residue, and for this, it is in the core, it tends to look at the helical residues that are pointing towards the beta sheet as well. And that ended up being a pattern that is throughout a lot of, it just is something that emerges in different types of structures as well. The preprint has more information on all beta or all alpha as well. But for time's sake, what is observed is that if a, so in this case, with the regularization, if essentially the first shell ends up being randomly masked, then the second shell residues that are bulkier are always the first to be paid any attention. So it ends up being the most important to keep track of bulky residues, and the model picks up this pattern And we can relate it to, the sidechain surface area, making it easier to, or just becoming crucially more important for the predictions. So before the rest of it, I'll just mention that the model is trained for single chain predictions, and it's compared to protein MPNN on the CATH 4.2 set. So the protein MPNN model mentioned in this work is also trained on the same splits without backbone noise. And this also relates back to the paper I mentioned earlier from John Ingram. And yeah, and the loss is just the standard categorical cross entropy. So before specific benchmark results, I also just want to show what the sequence embeddings look like at different layers. So this is for a fully trained model. And I mentioned that there were 8 total layers. So this would be the final layer. And all of these layers, for example, have edge updates followed by them, except for this one. And past this layer 8, we have the sequence. So in here, and I'm choosing to skip some of the layers out of just for space, but what is interesting to look at is even in the earliest layers, what they've learned that is very distinct and is possible to visualize is, so there's this pink thing and this green thing. And basically the pink is proline, and that light green color is glycine. So in every layer, it is essentially always very distinct where proline and glycine are in terms of the embedding space. And it's very easy to visualize that type of thing because it just separates a lot from every other residue. So that makes sense due to their physicochemical properties and what makes the actual types of the amino acids distinct in what they are. But what we also see is that around layer 6, especially, all of the identities are very distinct and possible to somewhat visualize separately. And this is just saturated at layer 8, where you see a breakdown of what you might expect just from what we know about amino acids in general. But yeah, so what is also important to note here is that there isn't an equal representation for the amino acid types because they're not going to, there isn't the same number of, valines as methionines in general. So this is just a mere visualization of what was possible to do with the test split. And so in comparison to protein MPNN, so on the left here is just a head-to-head native sequence recovery plot. So we have Frame2seq and protein MPNN sequences based off of, or predictions for the test set backbones. And what we see is that, first of all, they tend to do well on the same targets and poorly on the same targets. So we see an average improvement, which works out to about 2% in terms of Frame2seq's performance compared to protein MPNN. And then in terms of why they do similarly on similar targets, is possible to explain by data. So what those targets look like, and they're also learning on the same splits. So that sort of explains that they're just learning from the same available data. And then when we looked over what sequence recovery looks like for different lengths, so this is for the protein length, we didn't necessarily see a length bin that was that stood out, but it was that the 2% recovery was over all lengths between the two models. So Frame2seq would be this blue one, and protein MPNN is the orange. And
can I ask a quick question about the previous slide?
Yeah.
Why is it that they both tend to perform worse at lower length? Is that just less represented in their training sets, or?
yes, there are less. Let me see if I have a. So you can sort of see sequence length distribution over the different splits here. So there's training data for, below 100. But it is a lot less represented than compared to all of these that are way longer. And then I don't know if you can see the test and the validation well here, but yeah, so it can reflect the overall length distribution, which it most certainly does. And in terms of what is, I guess, available for the model, you can think that there is less restriction, geometrically speaking, when there's a shorter protein. So the backbone itself might be harder to design, or there might be the sequence space available for that backbone might look different.
Okay, thank you. That's really helpful.
So yeah, I'll also just answer a question that I just see in the chat, which is, did you do additional comparisons to ESM-IF? Have there been any interesting comparisons before, or do you have any additional thoughts on this? So for this, we just wanted to keep it apples to apples. So ESM-IF is not trained on the same splits. And so just out of fairness, it is compared to a model that is. And also we wanted to see in comparison to an experimentally validated model, which protein MPNN is extensively validated and performs very well. Okay, so yeah, jumping to a quick burial or native sequence recovery as a function of burial. So in here, we're looking at average beta carbon distances for A-close residues. And essentially over here on the left side is going to be more buried positions. And then as you go here is essentially surface positions. So what we see is that there's a similar trend, which makes sense. So it's easier to recover buried positions in general. Again, there's more information to just tease it apart from. So there is more geometrical context for a buried position than a surface one. Also, surface positions can adapt different amino acid types depending on what the surrounding biological context is. But in this case, what we see once again over the test set is that there isn't necessarily improvement over protein MPNN at these buried positions, but over some of the surface positions, there is some improvement here. And this can be possibly tied to learning something more specific about the surface with that restricted capacity to pay attention as well. And here are some quick results in terms of how both of these models perform with AlphaFold 2, or in terms of how they essentially how their AlphaFold 2 predictions compare against each other. And so in this case, so A and B are going to be when provided no MSAs and only a single sequence. So it's either a Frame2seq predicted sequence or a protein MPNN predicted sequence. And what we're looking at is the accuracy of the AlphaFold prediction. So the output from AlphaFold versus the confidence between the two models. And what we see is that they perform similarly. So I have there is a table on this and more information, but essentially they perform similarly. And this turns out to be the case with MSA. You can see in terms of this accuracy as well as the confidence that giving MSA spikes things quite a bit. So it's easier to predict or it's easier for AlphaFold to reach higher PLDDT when the Frame2seq and the protein MPNN sequences are predicted with MSAs. So that makes sense. So both of the models' output sequences sort of resemble some of the natural proteins, and there's a way to align them, with their accuracy. That makes sense. And in terms of how these sort of fare against the native sequences is also another comparison. I'm not showing it here, but it turns out that both of the models do better than the native when there are no MSAs provided, and neither of them do better than the native when an MSA is provided, which also makes sense. And something else that we looked at, and I said I was going to mention, is sort of the scores that can come out of Frame2seq. And so in this case, it will be in terms of I'll just call it the pseudolog likelihood, but there's also an example of how to use Frame2seq with just scoring or negative pseudolog likelihood. And in this case, so on the x-axis, essentially what we have as we go toward the right here is going to be a more favorable score. So according to Frame2seq, the sequence likelihood is increasing as we go here. And then native sequence recovery increasing on the y-axis. So in this comparison, what we're looking at is if you pass the true backbone and the true sequence and ask Frame2seq to score that, the true or native sequence, what you would get. And then with the blue scatters here, it's going to be for the Frame2seq predicted sequence and the true backbone, what is the overall score. And essentially both of these correlate very well with the native sequence recovery. So the model is aware that it isn't able to, either recover quite as well or recover very well, so that it scores them favorably or not. And then another thing to observe here is that Frame2seq prefers its own predictions over the native sequences, to which I think there are possibly multiple explanations. But the first one is, of course, this model isn't perfect and it is biased towards things. And it isn't going to do well in terms of every single target necessarily. And another explanation is not all native sequences are optimized to their fullest extent. So from what is available and what is collected in terms of that, structural biology data, is it's possible to find solutions that go beyond this native sequence and are scored favorably because maybe it is a more optimized version of that sequence for the backbone, for the exact same backbone. And it's just sort of a case by case, but possibly other explanations as well. And then this is essentially a binary way of showing it at the residue level. So in this case, correct just means native sequence was recovered. Incorrect is it wasn't. So between all of the predictions, whether they're correct or not, if you look at the residue's own score, what we see is that it's also very easy to for Frame2seq to tell apart when it has gotten a single position correct or incorrect, which is also very useful if you're sort of using it to figure out just by a quick scoring pass which residue positions are most likely to be of good quality and you don't want to change them versus the other scenario. Okay, so this part is a little bit abstract, but let's imagine this is the continuous protein space, and let's just imagine some blobs. So for these, the way I'll explain them is, let's say these are based off of structural spaces. So a single blob is bounded by belonging to one structural, let's say, family or specific functional family. And within this, based on the colors, so essentially the brighter it gets is going to be, it has very high homology to every other member in this cluster. And essentially the bounds or the outskirts of the family, the sequence is going to look more different than the average member from the family. So that's just an abstract way of thinking about what the sequence space would have been if conditioned on the structure. And so one thing that has been done and that works well for certain applications, directed evolution or other types of experimental flows, where if you chose this family and wanted to expand on the sequence, maybe with directed evolution, you can sort of step outside of what's possible, but sort of reach around the boundary here. And the question was, well, what if you really wanted to step outside and really far as you could get from all of the sequences that belong to this cluster, but still belong to it because you have the same structure. So if we did this randomly, of course, we would get a bunch of misfolded proteins. That space is vast. But if we did this, maybe guided by some type of model, in this case, Frame2seq, our question was, what sequence identity can we achieve where we're staying in the near identical structure space, but the sequence space is completely different, or as different as we could get to. So to answer this, we set out to design two de novo backbones, or design sequences onto two de novo backbones. In this case, this is the 2LV8 backbone I mentioned earlier, but also the historic top 7 fold right here. And our design process was give the backbones to Frame2seq, generate a bunch of sequences, use AlphaFold as a filter, where we had various RMSD and PLDT cutoffs for selecting higher quality designs. And so for these two backbones, again, we set out to have as low sequence identity as possible to the starting de novo proteins, but still look like them. And designing for low sequence identity with Frame2seq, we got a bunch of designs and picking the best ones with, PLDT, etc. Let's just talk about sets of 8. So 8 designs for 2LV8, 8 designs for top 7. And so what you're looking at right here actually is a 0% top 7 design. And in this case, we had sampled up to 50%, but you can see that we really aimed for some of the lower identity ranges as well. And before I talk about experiments, I'll just motivate this a little bit further. But when searched against non-redundant databases, as well as the PDB, in terms of NIH BLAST local alignment search, we see that these sequences have 0 hits. So they don't match anything. And it obviously doesn't match its starting backbone, but there also isn't necessarily a design out there that these look like. And then for everything else, around these identities for different backbones, we do get the original match. So the first one is always the original protein that these came from. So, we can say around above 20% identity, it's going to start looking like the original top 7. So, yeah, Frame2seq designs stable, soluble, monomeric, and folded proteins, but what does it actually mean? So for the set of 8 onto the 2LV8 backbone, it turned out that we had a bunch of proteins expressed, and so if they were soluble, they were monomeric and folded, and most of the time they didn't melt up to 95 degrees Celsius. And for this, essentially we consider it successful if it was soluble, monomeric, and folded. And the melting temperature doesn't really matter, and we only had one protein that melted anyway. So I'll tell you what identities we tested for this, and the successful ones are colored in. So what you can see is we tried this range, nothing below 10% worked. But we had something here, so there was a 19%, etc. And okay, let's look at top 7. So for top 7, once again, 8 designs and a little bit lower success rate compared to the 2LV8. But essentially, if you look at what's successful, we see this. Okay, so we got some proteins in these identity bins, nothing below 10% worked. And at this point, we thought, okay, so clearly the challenge here is that you can't get anything to be below 10% and be stable, soluble, monomeric, folded. And that was until we tried this a 3rd time. So there's a new set, and there are 10 designs in this set. And we got a 60% success rate with this. So once again, if we look at what is successful for these 10 designs, well, we had a focused run for specifically, the below 10% and the 12% and 14%. And we actually had 0% that worked. And then there's a 5% that worked. And then there's also the 7% that worked. So this was for the top 7 backbone, and I'll just highlight a few of the experimental results here. But essentially for the 0% and the 5.4%, you can see the size exclusion chromatography trace here, suggesting that the proteins are monomeric. And then this is the circular dichroism, where you can see the signal for which would be expected for alpha beta protein, and it actually matches the original top 7 CD signal well in both of these cases. And then in terms of melting, you can see that they do not melt. So we were very encouraged by the 0% top 7, and for the rest of the presentation, I'll just call it top 0. But we were excited about this, and to take a look at it further with work from Mark Kelly and Amy Guo, we essentially looked at the HSQC for top 0. So for this protein, first of all, we can always compare it to the top 7 HSQC. And before I talk about that, I'll just say essentially past this or to the left of this 9 ppm and here, this is sort of representative of beta sheet being present in the protein. So there is beta sheet conformation, and there's also alpha helical conformation, and that's what we would have expected from the peaks from HSQC. And also comparing this dispersion to the top 7 HSQC from Brian Coleman and others' paper, what we see is that they actually have a pretty good match. So we have all of this data to suggest that structurally speaking, we're staying within very close structural space of top 7, but obviously we have achieved a 0% sequence identity. And this is another way of showing the sequence. Given the top 7 sequence here, or the amino acids, and then predicted amino acids, so it's just going to be top 0's amino acid. There is nothing in the diagonal because this suggests an identity match. So every position here is substituted to something else when it's designed with Frame2seq. And, we do see, so this is a valine to isoleucine, so there's a number of occurrences of what you would think is a trivial substitution. But among other positions, you also have very non-trivial substitutions where the model is essentially restricted to finding a solution that isn't the native. And so to do that, Frame2seq actually just given top 7 recovers it at about 50+%, maybe 55%. So to get it to predict sequences that look like top 0 is also it's some work because the model doesn't like to reach such a sequence identity, but it turns out that it does a good job at it. So we could filter and find solutions that seem to make sense and could experimentally validate them. So potentially expanding further in terms of the novel design over the sequence space and stepping outside of what might be common representations or more homology seen in either natural proteins or specific functions in nature is definitely interesting. But something else that is potentially interesting and could be explored is taking a look at 2 specific structures and then finding a sequence that matches both of them maybe equally well or somewhere in between, such that we have multiple conformations and we can design for essentially a protein switch where the conformational states themselves are accessible with the same sequence. And through this type of design, maybe we can score and find some type of solution and guide that space, since once again, the space is vast. And this figure essentially showing where we can design de novo proteins and step outside of the native and what direct evolution makes possible. It's also interesting to do this for proteins that where their function is not necessarily folding, but something maybe a little bit more complicated. So with all of that, in this work, we propose Frame2seq, which is an IP-based single pass, state-of-the-art fixed backbone design method, which we find to reach or achieve high or competitive accuracies, and also in terms of inference speed, it is pretty fast when ran over GPUs, especially. And we find that with this method, we can design for novel sequences such that we're reaching undetectably low sequence similarity to the starting backbone. And so some next steps include evaluation of a Frame2seq multimer model. So as I've mentioned, this model is single chain, and expanding to different folds and families. So I've also somewhat mentioned this, but finding where to design novel proteins where they also have other very interesting or biologically meaningful functions, and higher throughput experimentation in general. So I'd like to acknowledge my lab and all of the collaborators and co-authors. So Tanja, Kortemme, my advisor, and Kosuke, Eleanor, Amy, and also Mark Kelly. And so Frame2seq is open sourced and available for use. You can pip install Frame2seq, or the code is also on GitHub, and it should be able to, it should be very clear how you can, score with it and design with it. And hopefully you check it out, but let me know if there are any other questions.
Let's thank Deniz. Really wonderful, clear talk. Are there any questions?
I have a question, if that's all right. First, really nice talk. This is really cool, especially like the top 0 result. My question is, so when you were generating the sequences for the top 7 and Rossmann folds, I saw you did like AlphaFold screening step. Just to confirm, that was in like single sequence mode with no MSA?
Yeah. So yep.
Okay, so I'm wondering if you would do the same in the case where you're keeping part of the sequence fixed, in which case part of your protein would be like a sequence from a natural protein and the other part would be designed. Would it be equally valid to use AlphaFold with no MSA, considering that like you really wouldn't want to use an MSA for the design part, but for the natural protein part, not sure how well AlphaFold would work in single sequence mode. So what AlphaFold settings would you use in that case?
Yeah, that's a really good question. That's actually something that we played around with a little bit, not for these proteins, but for something else. And the MSA signal is very strong with AlphaFold. So even if you have, let's say, some poor designs with Frame2seq, they can look good just by, matching enough to have that deep MSA for a natural protein.
Right.
So I think what you're suggesting with maybe taking out the design portion, or weakening the signal in some type of way where you're modifying some of the MSA, either making it shallower or just like getting rid of some of the alignment, could work. I personally think that a higher confidence cutoff for when MSA is used is probably a good idea. Also, I think running in parallel with templates just to essentially eliminate things based off of either lower confidence and accuracy due to template or due to the MSA run sets you up for maybe higher success for challenging design cases.
Okay, thank you very much.
Thanks for asking.
More questions?
I have a question. Well, I guess more of a clarification. I'm having a hard time understanding whether, so are van der Waals interactions and other electrostatics taken into account? So the interaction between residues, is that taken into account when going from the protein backbone to sequence prediction?
The quick answer would probably be no. But some other answer is, the interactions essentially come from certain geometric orientations and restraint. So the coordinates themselves essentially, let's say they obey a specific physical interaction, then that pattern is possible to pick up for the model. As to if it's explicitly training for some type of physical objective, or for example, van der Waals interactions, the answer is no. But that doesn't mean it hasn't learned it. So there would be, quick ways to check to what extent it seems to match, physical interactions we would expect. But yeah, it isn't trained to learn a physical interaction. Yeah.
Okay, thank you.
More questions?
I'd like to ask a question about the UMAP projections.
Yes.
Awesome talk, by the way.
Thank you.
So in layer 2, do you know like what the distinct two like blobs might represent? Would it be like secondary structure?
Yeah, so I was interested in attempting to figure out what the two blobs were. I never did. I also had a similar thought to you that, okay, this layer is learning something about the structure where it's possible to separate them, but it doesn't relate to anything in residue identity because it looks like noise, just like fuzzball. But they're so distinct. Yeah, so it is interesting. I haven't done the work to actually figure out what's up with it, but yeah, what you're saying is that's exactly the thought I had. It has to do something with structure that doesn't restrict the amino acid type selection, I guess.
Right.
So no idea.
It's really cool. Thank you for showing me.
Thank you.
More questions?
I've got a question. So I was wondering if you could speak a little bit more on how you generated those low sequence identity sequences, especially like, for example, for top 7, you said that natively Frame2seq gets a roughly 50% accuracy, or sequence recovery, sorry. So I was wondering, could you speak a little bit more on how you coerced your model to give you these low sequence identity sequences?
Yeah. So for these, and for anything that went into experiments, I should first say cysteines were always omitted from the vocabulary. And further than that, yeah, as you're mentioning, it can recover way more positions. So for every single position during inference, sampling from distribution, basically the native amino acid type was restricted. So the logits were essentially set to negative infinity so that it couldn't possibly sample something that matches, for example, the top 7 sequence. So for 0%, all of that was blocked. So every single position, the native was blocked. For some of the other ones, so for example, 23%, it was just randomly picked which positions would be blocked. And then just sampling got us somewhere, between certain ranges. So yeah, we tried to make it as random as possible for everything above 0, essentially.
Perfect, that makes sense. Thanks.
Yeah, thank you.
Really beautiful work. So in the pre-AlphaFold era, sequence recovery task was done, say, with Rosetta. And we were getting, much worse recovery, and we were sort of fighting for that last percent. And now we're kind of in a totally different regime where we're getting it so much better. I'm curious if you could, thinking about this, let us know where we're going from here. Do you think that maybe with lots more data, we can really continue to improve the sequence recovery even further? Or do we need sort of a new benchmark or a new goal for what we should be striving to achieve? You could think that maybe native sequence recovery is not the end all. Maybe there's like the actual contribution to stability might be sort of more fundamental because many native proteins are only marginally stable. And if we try to optimize for that, then we'll only get marginally stable proteins at the end of the day. So I'm curious on sort of how far can we push this and what are sort of the ingredients that you think we might need to get there?
Yeah, that's a really good point. I also think that native sequence recovery is for, I guess, current state of the art. It made sense to benchmark it and, learn for it for making some of these goals possible. But also, I think us doing the low identity work is showing that it's not necessarily meaningful. So that's something else that I wanted to come across with the paper, that because nobody wants 100% native sequence recovery, right? That's not a good model if it got to a point where it was just like error free. So with acknowledging all of the bias in, both the data and what we collect the data for and what the model is learning and how it gets its own bias, I think it will be increasingly more important to come up with other types of benchmarks that are perhaps more functional. So thermostability, like you're saying, or other ways of keeping track of some function, or some way to incorporate different types of representations. So maybe stepping outside the boundary of structure to learn something besides, just a loss term on essentially native sequence recovery. Yeah, I think in the future, it will hopefully have a lot more to do with function, but we're also in a place where you can't really have a training goal for sequence design without exploring the native sequence recovery goal, because at least that's one data point we know, okay, that protein folded. So it's really hard, but yeah.
And I guess where are we going to get the next like tranche of data to do this? So have we exhausted cath? Do we have to go into, AlphaFold predicted structures with like new sequences or metagenomics? Like where are we going to get the new bits of data to train this?
Yeah, I mean, those sound good. I think AlphaFold DB is single chain as well, but maybe if we have more that type of data, but for multi-chain, it could enable other types of advances, especially for, functional interfaces and such, which we have not explored yet.
More questions?
I have one dovetailing off of that. If you were to train on AlphaFold DB, for instance, where disordered sequences are represented, albeit not by very meaningful conformations, do you think a model like this would ever be able to meaningfully generate disordered sequences as well as predicting sequences that fold to a specific structure? Or would it just generate kind of like an artifact in those regions?
Yeah,
right?
that's
Because AlphaFold DB has disordered regions there, but they just kind of look like noodles.
Yeah, I don't think it would do well. I'm not super familiar with what the sequences look like for disordered regions in general, but if there was something it could pick up on it, and it was prominent enough in the data, I think something that could go wrong, I guess, is for potential unstructured regions and other types of structures. It could try to essentially design for that IDP signature. Yeah, I'm not sure. Yeah, I would have to look at the data, but yeah. Sounds hard. Yep.
Just curious. Thank you.
Yeah, thanks for asking.
More questions? So we had Rohit come and tell us about the all atom version, and I guess with the next version of the protein MPNN, there's this idea of getting away from like just native sequences. So like with cofactors or post-translational modifications or non-canonicals. What is there something fundamentally prohibitive in your model that would make that difficult, or is it sort of a similar idea? You just increase the vocabulary and you can do as good as you can as long as there's data.
Yeah, I think it's definitely possible to have a workflow that has the all atom idea and representation for the predictions. I think in terms of what context it's for, It would just have to be carefully thought out in terms of where to incorporate and what kind of data is available, depending on the context. Yeah, so it probably wouldn't be like an all doing model, but start from, whatever the function of interest. So if it's ligand binding, then maybe a starting point in some sense.
I guess maybe very specifically, do you see sort of phantom metals show up in your sequence designs?
Okay, yeah, there is something interesting. I'm not sure, whether to overinterpret it or not, but there is a part of so for top 0, it turned out there is a portion of the sequence that recognizes nickel. We haven't really explored it, and it wasn't meant to do that. I don't know if I would be aware of any other way to keep track of it. This I only know because Amy told me, but yeah, so it ended up having certain things in a row in the sequence. In the primary structure, it made sense where it was creating some context for metal binding, possibly. Yeah, so
There's these tools like AlphaFill that try to come in and place metals and ligands and things into AlphaFold structures. I'm curious if you ran AlphaFill on your sequences that you designed, if you would get more or less of those types of, placements of things.
yeah, that's an interesting idea. I guess I could take all of the AlphaFold predicted structures and just see if there is anything there.
Any other questions? Okay, well, let's thank Denise one more time. Really fantastic, really fun to see. Amazing that we're pushing the state of the art so far. It opens up a lot of really exciting applications.
Thank you so much. This was a lot of fun.
Okay, see you next time.